Hello and welcome back. In this third video in our segment on machine learning methods, on supervised machine learning methods, we're going to delve into introducing principal component analysis, often just referred to as PCA. So the idea behind principal component analysis, of course, is just like those other methods, is to reveal some structure in your data, some patterns in your data that might be indicative of something you want to dig into further, some underlying biological mechanism, for example. So let's start at the basics and consider an example where we've got a bunch of cells here that we're viewing under a microscope, and they all look kind of the same. Even though we suspect there are differences about them, we can't really see it in traditional ways. So this might be one cell type, for example, this another, and maybe we've got a tumor cell type in here, something we really want to find out about what makes it different, for example. So unfortunately, we can't view these differences directly by traditional approaches. So one thing we could do is we could look at the amount of messenger RNA in each of these cells and identify by that approach which genes are active and which ones are less active at a given time. And then the idea being that those that have similar gene expression patterns are probably doing similar things with their gene products and are likely similar types of cells, for example. So the kind of data we'll get from this, and we'll actually see this in the next segment of our course where we calculate this kind of data ourselves, is we'll get a table like this, right, where we've got genes down the rows, and then we've got the columns, or our variables here are different samples, right? So cell type 1, cell type 2, cell type 3, etc. And we've got different values in them. For example, the counts for these different genes. So what do? Well, here, each column, as I've said, shows how much of a gene is transcribed in each cell type. And let's start simply. Let's consider just two cell types, and what we could do is we could just plot one against the other. We could take these as the axis of a plot. So here I've done that. I've taken cell 1 and cell 2 and I've plotted the genes on this scatter plot. And we could see here some interesting things about the data. When it's high in this gene 1 here, when it's high in cell type 1, it's low in cell type 2. And conversely, this gene right at the bottom of the list, gene 9, when it's low in cell 1, it's actually high in cell 2 type. So we can postulate, based on this pattern, that these have this inverse correlation that suggests that they may be doing quite different things with their genes in these cells, and thus the cells might be of different types, for example. Now, let's imagine we have three different cells to consider here. And we've already done cell 1 and cell 2, but we have to now consider cell 3 as well. So we have to consider a plot perhaps of cell 1 versus cell 3, something like this. And here we're seeing a positive correlation in this case. So in contrast to those first two cell types, this suggests when the genes are up in cell 1, they're also up in cell 3. When they're down in cell 1, they're also down in cell 3, suggesting this positive correlation, suggesting that they're doing similar things, perhaps similar cell types in this case. And then we'd also have to do cell 2 and cell 3 here again, and we might see this again, this inverse correlation, suggesting that they're doing different things. And another way to do this, of course, is we could plot a crazy kind of 3D plot, right? So that third cell dimension is disappearing into the board here behind me, and the points that are smaller are further off along that cell 3 line. And that's okay, but what if we have more, right? 4, 5, 6, many dimensions like we typically have in these experiments? Well, we could try and plot lots and lots of little 2D plots like we were doing and look at these correlations to try and make sense of them. Or we could try and draw some crazy ass graph with many axes and dimensions, which just is pretty impossible to interpret at the end of the day and would make your head kind of explode if you try and think about this too much in this kind of plot. Well, this is where PCA can come to the rescue. It'll stop our heads exploding, right? It's going to take this results, our big matrix, our multivariate matrix, multiple variables here that we're looking at, multiple columns here for many different rows or genes in this case, and convert them into something we can more easily grasp and interpret and visualize, like a little 2D plot where we're plotting PC1 versus PC2, and we see the points in them are the actual cell types. Okay, so this would suggest that all these cells that are lie close to each other, they're correlated with each other and doing similar things with their gene expression patterns, as are these other two groups of cells, suggesting we have maybe three different types of cells in our original setup. Okay, so if we feed that back to our original data, we would see that indeed there are 3 different cell types as revealed by principal component analysis results here. Okay, so some key points before we move on to talk about how this works. The PCs, or the new plot axis, these principal components that I'm plotting here of PC1 versus PC2, they're ordered by their importance. PC1 is always more important than PC2, which is in turn more important than PC3, which is in turn more important than those lower number or those higher number PCs 4, 5, 6 in order. So they're ordered by actually how much variance they capture on the original data set. And PC1 is always more important than PC2. Okay, so in that case, what it means is, if we look at our original plot, that red grouping of cell types compared to the blue and the yellow, they're more dissimilar from each other, the red versus the blue, than, for example, the blue and the yellow are, because PC1 is more important. It's capturing more of the variance, so there's more spread captured in that PC and there, so it's going to show us that those are more dissimilar in this case. And you'll often see in the literature, when people plot these kind of graphs in their publications and whatnot, they'll have perhaps the proportion of variance captured along a given principal component, a new axis of our data set here in parentheses. Like here I've placed 44% along PC1, indicating that 44% of the original variance or spread in the data is captured by this new axis, this new PC1. And here only 11% is captured in PC2. If we had a plot of PC3, it would be a smaller number again. So in this case, PC1 captures much more of the variance, in fact 4 times more of the original variance than PC2. It's 4 times more important, even though we plotted on these axes with equal spacing. For example, the spread and the importance of this PC1 is more. Okay, that's an important point to consider. So we also get another very useful thing out of a typical PC analysis. We get eigenvalues as well as those principal components. You'll hear people refer to the PCs as eigenvectors. Those are the new axis. Okay, well, people call them PCs, principal components, or eigenvectors. And we also get a second thing, these eigenvalues. They actually detail the amount of variance that's captured in each eigenvector or each PC, and that's how they're ordered. Again, PC1 has most, as shown here in the plot, PC2 less, etc. So this little plot that I'm showing here, it's a result of analysis of some gene expression data where we have wild type and a gene knockout experiment here. We're looking at a number of samples or replicates, biological replicates for each condition here. Red is the wild type, blue is the knockouts. And we can see that PC1 is really splitting those two based on the knockout or non-knockout, the wild type or knockout. And it's capturing most of the variance, actually a huge proportion, 91% of the variance. So this result tells us that the gene expression in these samples is very different, and it's characteristic of either wild type or knockout. And we'll often use this as kind of a first pass or a first filter to see if the perturbation we're interested in, or the drug that we're testing, or the knockouts or the mutants that we're examining really do give consistent differences. In this case, this result, they certainly do, and it's worth digging into further. Now, another cool thing that we can get out of these principal component results is a quantitative report of how the original variables, that's the original genes in our case here, contribute to these new principal components, these new vectors that we're using to plot our data. Okay, so in other words, as I'm writing here, which is the most important genes for splitting, for example, the wild type and the knockout. These are often called the loadings, and we can plot these as we'll see in our hands-on session. We can plot the loadings to see which of our original genes contribute most to this major spread or major split that we see along these interesting components that we might want to do. Okay, so we're now going to do a little hands-on time. We're going to jump over to our class website, and please visit our PCA web app that will walk you through some of the intuition that's involved in understanding these new components. It takes you up from 2 dimensions to 3 dimensions, up to a 17-dimensional data set, and then some gene expression data with many dimensions. And we see how principal component analysis can add value to our interpretation of the underlying structure in all of those data sets. And then once you've done that, I'm going to come back and we'll talk about how to actually do this in R. Okay, how do we actually do principal component analysis in R? Well, there are quite a few different packages that will do principal component analysis. There's a number of bioconductor packages, for example, and even on CRAN, there's quite a few packages that are devoted to doing PCA on particular data types, like Bio3D for structures and sequence data, etc. And we're going to focus on here. We're going to focus on the base R implementation in pt1. That's the base function that's available right out of the box in R to do PCA. There's a little bit more work to do with it than some of the add-on packages where the plotting methods are often easier, for example. But if we know how to use this one, we'll know how to use them all, really. Okay, it's one of the harder ones to interpret and make our figures that we want to put in our papers. But if we can figure that out, we'll be good to go with pretty much all of them. Okay, so here I'm reading in some example data. This with read CSV, we'll do this too in our hands-on section. This is gene expression data. Note that the samples here are columns and the genes are the rows, just as we would normally see in this type of data. Then we're going to call pt1 function on this data set. The base R function here, it wants actually the transpose of this. It wants the samples to be the rows and the genes to be the columns. So we're calling the little t transpose function on our input data. And we're also setting the optional scale argument here to true. We'll talk about that, what that does in a moment. We look at what the results are given here, what attributes are in this PCA object. It's got a number of things that are returned, including the standard deviation, which is obviously related to the variance. We can square one and take the square root of one and get the other. And then we've also got this thing called X. What are these things? So this thing, $X, that we would access here, if we called our result PCA, we'd write PCA $X. It contains the actual principal components, the PCs. And we can use this thing to draw our graph. It's the scores of our original variables along these PCs. So if we take the first two columns of this thing, of PCA $X, we'll get our little score, our score plot, our PC1 versus PC2 plot. So we can do that like here. PCA $X, X is the first column, PCA $2, X is the second column, and we'll get our base, it's not very pretty yet, but this is base R graphics PCA plot, result plot. We could look at the variance along given principal components. We take the square of that standard deviation to get the variance. We could do the cumulative variance, and we could see that PC1 in this case accounts for over 90% of the original variance. So it's really where all the action is in this data set. PC2 is almost 3%, PC3 is under 2%, and then we've got 1.3, etc. So they're ordered again by how much variance they capture in the data set. So I'll produce a little bar plot here of that variance, and we see again PC1 is by far the most dominant PC in this result. And what that's suggesting, or what it's indicating here, is that this spread is splitting these things we want to know. Is what's captured here? What are these samples? What are they doing? So they're actually, as we can see, they're wild type and knockout. The red samples and the blue samples here are being split very clearly along principal component 1 in this case. Okay, so I want you to go to our hands-on sheet and we'll actually work through this example ourselves. We're going to go and do PCA of that data set that we just talked through here, but we're also going to do it of some more traditional, kind of conventional data sets that might be easier to understand, including making fun of things that people eat around Ireland and the United Kingdom and Europe, and seeing if we can see structure in that sort of data. We'll see that principal component analysis is a ubiquitous kind of method for giving us new insight and useful insight into finding structure in all sorts of different types of data, numeric data. The main objectives of this method are to reduce the dimensionality. So rather than consider all the dimensions, all those things that we're measuring, we can reduce it down to something much more manageable. We don't have to make that crazy ass plot with all those dimensions, but we can take the results of PCA and view a much lower dimensional representation. 2 or 3 dimensions usually is sufficient for the kinds of data that we're analyzing, and allow us to plot that up and see patterns in our data, to see if there's structure in our data. We often use PCA as a kind of first filter approach to select the most useful variables to go forward for other machine learning methods, for clustering, for example, in that PC basis set. And we'll do that in our hands-on session with the biopsy data of cancer cells, breast cancer cells. And we do it PCA to identify outliers as well, of course. If there's one point that's way off further from everything else, as you'll see in our UK foods data set, that's an outlier. There's something different about that country versus others. There's something different about that experiment that you've run that you might want to dig into further. Maybe it's something interesting, or maybe it indicates a problem with the procedure that you used for that one example. Okay, with that, I'm going to leave it there for now, and in our next video we're going to dig a little bit more into principal component analysis, explain how it works, as well as talk about some of the related methods that have a similar aim that are becoming more and more popular in this area, namely t-SNE and UMAP. We use these a lot on, for example, single-cell RNA-seq data. So join me then when we dig into more on these multivariate analysis approaches. Thank you.